All right, welcome to the Explainer. Look, if you are currently grinding through your prep for the MCAT, AP Biology, or your A-levels, I know just hearing the words Enzyme Kinetics or Michaelis-Menten probably makes your heart rate spike a little bit. It honestly feels like you're about to be buried under a massive mountain of confusing equations and weird graphs. And hey, it is a completely normal reaction to feel overwhelmed by this stuff. It's tricky, but I want you to just take a deep breath right now because today we are going to completely crush this topic. I promise you, we're going to build a foolproof, tactical guide to make these complex biochemical concepts crystal clear and honestly highly intuitive so you are totally ready for your upcoming exams. Here is the exact roadmap of what we're covering today. First, we'll look at the core reaction. Second, Vmax and Km. Third, the Lineweaver-Burk plot. Fourth, enzyme inhibition. And finally, we'll wrap up with the test day survival guide. So, let's set the stage here. Inside your body, at this very exact moment, there are literally thousands of chemical reactions happening every single second. So how do they all occur without just completely crashing into each other in total chaos? Well, the answer is that almost none of them could happen without enzymes. But enzymes don't just switch on and blindly run at full speed all the time. They speed up, they slow down, they get blocked, and sometimes they just get completely overwhelmed. They require an ultimate traffic controller. And understanding exactly how they manage that cellular traffic flow, well, that is exactly what Enzyme Kinetics is all about.
Okay, let's dive right into Section 1, the core reaction, understanding the foundation. Basically, enzyme kinetics is simply the study of how fast enzymes work and what actually affects that speed. But before we can measure the speed of anything, we have to know the steps of the reaction itself. It's kind of like a simple formula. Step by step, it goes like this. The enzyme, which we represent with an E, plus the substrate, S. And remember, the substrate is just the specific molecule the enzyme works on. They come together to form what's called an enzyme-substrate complex, or ES. Once they are locked together, the enzyme does its catalytic job, and that complex breaks down into the free enzyme, E, and your finished product, P. And the really cool part? That enzyme is now totally free to go grab another piece of substrate and do the whole thing all over again.
Now, if looking at those letters still feels a little bit abstract, I want you to think about it this way. Imagine the enzyme is a barista working at your local coffee shop. The substrate, that represents the incoming coffee orders from the customers. And the product is the finished, hot cup of coffee that gets handed across the counter. Now, if there's only one single customer walking into the shop, our barista handles it easily, right? But imagine it's 8 a.m. on a Tuesday. The morning rush hits. As more and more customers pile in, the coffee orders, the substrates start piling up too. The barista gets busier and busier, frantically pulling espresso shots and steaming milk, until at some point, they are working at their absolute maximum speed. They physically cannot go any faster, no matter how many more frustrated customers shout their orders at them. That physical speed limit is exactly how enzymes operate in your cells when they get totally flooded with substrate.
Moving on to Section 2, Vmax and Km, decoding the speed limits and affinity.
Alright, so the first absolutely crucial concept you need to lock down is Vmax. Vmax stands for maximum velocity, and it is exactly what it sounds like. It's the absolute fastest the enzyme could possibly work. It happens when every single active site on every single enzyme molecule is completely occupied with the substrate. Going back to our coffee shop analogy, Vmax is when every single barista in the shop is making a drink at the exact same time. There's absolutely no spare capacity. Every single hand is busy. Now, here is a massive point I want to emphasize heavily because examiners absolutely love to test this. Vmax only changes if you change the amount of enzyme. If you double the number of enzymes or hire more baristas, your overall Vmax doubles. But once all those active sites are full, adding more substrate does absolutely nothing. The baristas are already at maximum capacity. If you were to plot this out on a standard Michaelis-Menten graph where you have substrate concentration on the bottom x-axis and reaction speed on the side y-axis, you wouldn't just get a straight line that goes up forever. You get a curve that rises really quickly at first and then sharply levels off into a flat plateau. That flat plateau, that right there represents Vmax. The whole shape of that curve visually tells us that adding more substrate eventually just stops helping.
Okay, the second key number is Km, which is also known as the Michaelis constant. Now, I know this one confuses a lot of students, but let's define it really simply. If you look at that curved Michaelis-Menten graph we just visualized, Km is the specific substrate concentration on the x-axis that gets you exactly to half of Vmax. It's simply the amount of substrate you need to reach half of your maximum speed. But what does Km actually tell you biologically? Like, why do we care? Well, it tells you about the concept of affinity, which basically means how tightly the enzyme holds on to its substrate substrate. Think of it like a magnet. If you have a really, really strong magnet, it picks up a paper clip from far away effortlessly. That represents a low Km. You don't need much substrate to get things going, meaning a strong grip or high affinity. But if your magnet is super weak, you have to literally shove the paper clip right up against it to get it to stick. That represents a high Km, meaning a weak grip or low affinity.
To really make this stick, let's look at a classic real-world examiner's love, comparing two enzymes that both process glucose. We have hexokinase and glucokinase. Your muscle cells use hexokinase. Now, hexokinase has a low Km, which means it has high affinity. It works super efficiently even when your blood glucose is really low. And that makes perfect biological sense, right? Because your muscles need a steady, reliable supply of energy all the time, even if you haven't eaten for hours. On the other hand, your liver cells use glucokinase. Glucokinase has a high Km, meaning low affinity. It really only kicks into high gear when blood glucose is very high, like right after you eat a massive meal and your liver needs to store the excess. It is the exact same chemical reaction, but these inverse Km values serve totally different biological purposes in different parts of your body. It's actually brilliant when you think about it.
All right, Section 3, the Lineweaver-Burk plot. Flipping the script on graphs.
I know the name Lineweaver-Burk sounds intensely complicated, but stay with me. Instead of plotting a curved line like the standard Michaelis-Menten graph, the Lineweaver plot simply flips both axes. You're plotting 1 over velocity against 1 over substrate concentration. It's a double reciprocal plot. And the beautiful thing is, instead of a curve, you get a wonderfully straight line. Why do scientists use it? Because straight lines are just so much easier to read and mathematically extend. You really just need to look at the intercepts. I want you to mentally tattoo this strict rule on your brain right now for test day. The Y-intercept equals 1 over Vmax, and because it's an inverse fraction, a higher Y-intercept actually means a lower Vmax. Let me say that again. A higher point on the Y-axis means a lower maximum speed. Then look at the X-intercept. The X-intercept equals -1 over Km. So exactly where that line crosses the X-axis directly tells you your Km. Memorizing what these two specific intercepts represent is honestly the golden key to unlocking this entire topic.
Moving to Section 4, enzyme inhibition, when things get blocked.
Let's walk through the actual mechanics of enzyme inhibition, because you will absolutely see this on your exam. There are 3 main types. First, competitive inhibition. In this scenario, the inhibitor looks almost exactly like the normal substrate and literally competes for the exact same active site. It's kind of like jamming the wrong key into a lock. The right key can't get in until that wrong key is removed. Because they are competing, you need more substrate to eventually outcompete the inhibitor, so your Km goes up. But if you flood the system with enough substrate, you can eventually overwhelm the inhibitor and still reach your maximum speed, so Vmax stays the same. Next is non-competitive inhibition. Here, the inhibitor binds to a completely different spot, called an allosteric site, and it changes the enzyme's overall shape. Because it's not blocking the active site at all, the normal substrate can still bind just fine, so Km stays the same. But a fraction of your enzymes are essentially broken and distorted by this process, so your overall Vmax, your maximum speed, goes down. Finally, we have uncompetitive inhibition. This one is weird. The inhibitor only binds to the enzyme-substrate complex, basically locking it together after the substrate is already attached. The unique result here, both Km and Vmax go down by the exact same factor.
Now, speaking of uncompetitive inhibition, it has a very distinct visual signature that you need to know. Because both Vmax and Km decrease by the exact same ratio, the slope of the line doesn't change at all. So it creates perfectly parallel lines on a Lineweaver-Burk plot. If you are ever looking at a passage-based test question and you see a graph with parallel lines shift upwards, alarm bells should immediately be ringing in your head. Uncompetitive. This specific graph change is a classic standardized test trick question, and now you're literally never going to get it wrong.
So let's rapidly list off some real-world drug examples of inhibition that frequently pop up in MCAT and A-level passages. We want to completely bridge the gap between this textbook theory and real application. First up, statins. These are widely used to treat high cholesterol, and they are a perfect example of a competitive inhibitor. They physically block an enzyme in your liver called HMG-CoA reductase. Second, aspirin. Aspirin irreversibly inhibits COX enzymes to stop pain and inflammation in the body. And third, penicillin. Penicillin irreversibly inhibits the enzymes that bacteria use to build their cell walls. Without those walls, the bacteria just burst. Knowing these specific real-world examples gives you incredible context when you're reading through complex exam passages, because you instantly understand the actual biochemistry behind the medicine.
Finally, Section 5, the Test Day Survival Guide. What you need to know cold. All right, let's go through the progressive checklist of the 6 absolute must-know facts that you need to have totally locked down before you walk into that exam room. Step 1, master that foundational equation. Enzyme plus substrate goes to the ES complex, goes to enzyme plus product. Step 2, remember Vmax is maximum velocity, and remember that the Michaelis-Menten curve plateaus because velocity does not increase with more substrate once those enzymes are fully saturated. Step 3, Km represents affinity. Remember our magnet analogy. Low Km means high affinity. Step 4, know those Lineweaver-Burk intercepts. Y-intercept is 1 over Vmax, X-intercept is -1 over Km. Step 5, memorize the exact mechanics in that inhibition table. And Step 6, remember the real-world drug examples we just listed to firmly anchor your knowledge and reality.
Well, you've made it. You now have the keys to enzyme kinetics. So which exam question are you going to crush first? Think about it. In just a few minutes, we went from the basic barista coffee shop analogy, we decoded the speed limits of Vmax and Km, we visualized the curve, flipped the script with a Lineweaver-Burk plot, and completely mastered the 3 tricky types of inhibition. You have a brand new, highly effective toolkit. I highly encourage you to pull up some practice problems right now while all this structured information is super fresh in your mind, and apply exactly what you've learned. Put this knowledge to the test. Stay focused, trust the structure, and keep learning. You've got this.